Hey, thank you very much. I want to make sure. Yeah, Dr. Ho is there. And it is my pleasure to introduce today's speaker, Dr. Ming-Fen Ho from Mayo Clinic. Dr. Ho is Associate Professor in Psychiatry and Pharmacology, focusing on the neuropsychiatric disorder using advanced stem cell technology and patient-derived model. And the whole work is especially iPS cell-derived brain organoids. Also, we call the mini brain. She is very famous with the mini brain terminology and enable to study the human brain function and the drug response, including opioid and at the single cell level, and then providing new insight into the addiction and the therapeutic targeting. Her research target stem cell biology, neuroscience, and then precision medicine to advance the understanding and then treatment of the complex brain disorder. Everyone can ask the question directly, or your question can live in your chat box indirectly. Please join me in welcoming Dr. Ming-Fen Ho.
Thank you so much, Dr. Tim, for the introduction. I will share my slides. Go full screen.
Yep, great. And then you can make it to full screen.
So is this all right to you?
Perfect.
Okay.
Yeah.
So you can hear me all right as well. What I'd like to do for the next 20 to 25 minutes is to give you a broad overview of what we are doing in the laboratory using multi-omics and iPS-derived brain organoids as a cell model system to study neuropsychiatric disorders with a focus on substance use disorder. I have no conflict of interest to claim. So today we're going to talk about alcohol use disorder, which is the most common substance use disorder worldwide. There are 3 FDA-approved medications to treat alcohol use disorder. We call this AUD. Those drugs are underused. Less than 5% of the patients receive pharmacotherapy. The first drug I'm showing you here is disulfiram. That is a very old drug used to suppress alcohol metabolism. Naltrexone and acamprosate are anti-craving agents, which we can use these drugs to prevent relapse. We have been studying these anti-craving agents for years using iPS model system, but still there's so much we need to learn. Previously, I heard echo. So previously, we conducted a acamprosate clinical trial with 400 patients, all treated with acamprosate for 3 months. The purpose of this study was not to determine whether acamprosate works or not. The purpose of this study is to gather multi-omics for analysis that can help us to identify biomarkers for acamprosate treatment outcomes. So what did we collect? We have very comprehensive clinical phenotypes, including alcohol craving intensity, alcohol consumption, a series of rating scales related to mood, anxiety, etc., before and after treatment. We also generate a series of omics, including genomics, metabolomics, proteomics, and transcriptomics using blood-based assays. On the top of this, we have a panel of iPS cells for functional genomics. So the paper on the right, at least here, is the most recent publications using those multi-omics. So what I'm going to do for the next 10, 15 minutes is to highlight our most recent publications, focus on proteomics and genomics. That is a protein I highlight here, HSD11B1. This is showing you the clinical information for this particular study cohort. We have 400 patients with alcohol use disorder. 2/3 of patients are men, which is consistent with virtually any alcohol use disorder study ever conducted. 90% of the patients are white, since this is one single site study. We found baseline craving intensity as determined by PAX. I will explain this in just a minute. That is the most strong predictor for acamprosate treatment outcome. Relapse is defined as having one drink during the 3 months of acamprosate treatment. Non-relapse means those patients stay sober during the study course. We also study a series of comorbidities, including depression, anxiety, OCD, PTSD, but they don't seem to have much impact on acamprosate study. This study is conducted in psychiatry. So a lot of people ask me, Dr. Ho, how do you know your patient really take medication? If some of them did not take medication, this might be the reason why they relapse. And that was a fair question. So we studied the blood drug level. So the figure on the right showing you acamprosate blood drug level for patients who relapse or now relapse during the 3 months of acamprosate treatment. We found no significant difference between the two groups. And these data are telling us acamprosate works, but not for everyone. For some people, even they did take medication, but they don't have much effect at all. Why is that? Can we begin to individualize treatment? This is showing you what PAX is. PAX is a self-report rating scale for measuring alcohol craving intensity. There are 6 questions. Each question are asking people if they have any alcohol craving and how often is the craving and how strong the feeling is. So each question has a score from 0 to 6. The higher score, the higher craving intensity. But there is no cutoff to determine whether a certain number is defined as high craving intensity or the low number is low craving intensity. We don't have such thing. So this is a type of the phenotypes we study in psychiatry. I'd like to walk you through our research strategy that called proteomics-informed genomics. What we did here is to use baseline plasma samples for proteomics. That will help us to identify what proteins are associated with acamprosate treatment outcome measures. We have 4 primary outcome measures I listed here. I will walk you through in just a minute. Once we identify what proteins are associated with outcomes, we will perform genome-wide association studies for those proteins. We use protein concentration as a quantitative trait for genome-wide association study instead of using primary outcome measures for GWAS. There's a reason for this. Once we identify the SNP or genes associated with the protein concentration, we would replicate these findings using other GWAS. We will also functionally characterize those signals using IPS-based model system. So here is showing you our primary outcome measures, including relapse, yes or no, or heavy relapse, yes or no. So relapse is defined as having one drink during the 3 months of acamprosate treatment, whereas heavy relapse means 4 drinks a day for women or 5 drinks a day for men. We also study continuous variable, including time till relapse or time till heavy relapse. Those outcome measures are very similar, but it's still slightly different. This is showing you the proteomics data. We identify a number of proteins associated with these treatment outcomes. And there are 4 proteins I list here. They are overlap across all four outcome measures. We have studied a couple of these proteins, but today I really like to highlight HSD11B1. HSD11B1 is not only associated with a composite treatment outcome, it also associated with baseline craving intensity, which is a rating scale we use to measure alcohol craving. And we found positive correlation. The higher craving intensity, the higher level of HSD11B1, the p-value is very significant, 10 to the minus 7. I need to point it out. This study cohort is one of the largest composite studies ever conducted, and the only one in the world that we have multi-omics data for analysis. So this is a quite unique study cohort to pursue precision psychiatry. Once we have a signal, we like to pursue genome-wide association study, and here is the result. The GWAS result showed no significant signals that above genome-wide significant level, which is quite disappointed because there's no signal we can pursue functionally. So we take one step back, going back to the biology here. HSD11B1 is an enzyme highly expressed in the liver that convert inactive form of cortisol to cortisol. So cortisol is a stress hormone. It's an active form that can bind to glucocorticoid receptors and have downstream effect on a series of immune response. There's a recent brain MRI study looking at HSD11B1 signal in multiple brain regions from patients with alcohol use disorder or healthy controls. If you look at the figure down here, you can tell the rate is AUD show higher signal as compared to healthy controls across multiple brain regions. So this is telling us this enzyme is important and might have a critical role in the pathogenesis of alcohol use disorder and perhaps treatment outcome phenotypes. So once we figure this out, we go back to the cortisol. We use the same sample for cortisol measurement. And then we found cortisol concentration is positively correlated with baseline craving intensity. The effect size is small, but that's pretty normal in neuropsychiatric disorders. The p-value is significant. Once we observe this phenomenon, we move forward to genome-wide association study. So here is cortisol, the active form and the inactive form. We pursue genome-wide association study for the plasma concentration of these metabolites, and we found the same signal show up. Those signals are genome-wide significant. The p-value is 10 to the minus 9. The top hit SNP here is on chromosome 14. So if we blow this up, here is the top hit genetic variant, which we identify in that GWAS I just showed you. The p-value is 10 to the minus 9. The SNP genotype here is within a gene cluster on chromosome 14 in CBLN3 and KHNYN genes. And before we pursue this project, these genes we never heard of, but we have pursued functional genomics to demonstrate they do have some impact on cortisol concentration, which I will show you in just a minute. So once we have a signal, we found this signal also associated with composite treatment outcomes. The SNP genotype is associated with a composite treatment response. So if you look at the plot in the middle, the GG genotype is a common genotype that associates with beta composite treatment response, including tight tail relapse and tight tail heavy relapse, as compared to the heterozygous. The minor allele frequency is about 5% in this study cohort. So after we found this genetic variant genotype is associated with a compensate treatment response, we want to know what it does in the brain. As a first step, we create a construct that includes different genotypes of these genetic variants. And the GG allele is the common allele that shows higher transcriptional activity as compared to the variant genotype. That means that the genetic variant is functional. So this is at SNP level. And everything I just showed you is coming from blood-based assay. But alcohol use disorder is a complex brain disorder. So we take advantage of the iPS cell model from our patients. When patient came in, see a psychiatrist, give blood sample, sent to our laboratory, we make iPS cells. Once we have iPS cells, we can convert to virtually any cell types in human tissue. My lab focuses on neuropsychiatric disorders, so we make neurons, astrocytes, 3D brain organoids. This is a unique cell model system to study neuropsychiatric disorders. Why is that? Because one of the major challenges in our field is the lack of access to living human brain tissue. So this iPS model system allows us to recapitulate living human brain tissue to study the underlying biology. Each cell line we generate in the laboratory is coming from a real person. A real person, we know their treatment outcome. A real person, we have their EHR, and we also have really comprehensive rating scales we study in psychiatry. This makes this cell model system unique. So we create a mini brain study infrastructure at Mayo Clinic to support this effort, which composed of 5 major components, including recruitment, administrative, research, clinical, and computational. These 5 components are equally important. So currently, we focus on patients with addictions, including alcohol use disorder and opioid disorder. We also study mood disorders. And on the top of this, we have a subset of patients that are healthy control. Healthy control is defined as having no personal or family history of mental disorders, including substance use disorder. This is the brain organoids we generate in the laboratory. If you look at the up panel here, those brain organoids, we call this mini brain in a culture dish. They are really tiny at early stage, but visible by bare eyes. If you look at these brain organoids under microscope over time, you can tell the structure is very different when we differentiate those cells to 3D brain organoids. And these brain organoids also region specific. What I'm showing you here is 4 brain organoids. At 3 weeks, they are tiny, but visible by the eye. This is about 3 months. This is about 5 months. These brain organoids grow in size. So what we did in the laboratory is to use a bioreactor. We use a 3D printing material and put them together to culture these brain organoids. This is just showing you how this bioreactor works. We use this instrument to constantly stir the cultural media. And why do we do this? This is because brain organoids is a 3D culture, grow in size. So over time, the cells in the center, they don't have sufficient supply of oxygen. They don't have access to oxygen or cultural media. This is why we need to use this instrument to enhance the efficiency. Otherwise, over time, the cells in the center will become necrotic and eventually die. Once we have brain organoids, before we do any experiment, we characterize those cells. This is showing you how those brain organoids self-organize and self-assemble. Each color is a cell surface marker. SUX2 is a marker for progenitor cells. So you can begin to see how these cells self-organize and self-assemble. On the right, TUJ1 is a markers for neural cell type. So now we're back to science. We have IPS from our patient, we make brain organoids, and then we treat those organoids with ethanol or the two FDA-approved anti-quiving agents, including acamprosate and naltrexone. Then we send out the samples for single-cell sequencing. Each dot here is a cell from brain organoid. Each color represents a cell type. It can be glial cells, can be progenitor cells, glutamatergic neurons, or GABAergic neurons. When we look at those single-cell sequencing data, our focus is the two genes we identified during the GWAS for cortisol concentration. And we found KHNYN is highly expressed in the brain across all cell types in the brain organoids. But for CBLN3, the signal is really weak because it's not highly expressed. So we take advantage of those data looking at KHNYN gene expression. We found it's upregulated in the presence of ethanol, but the anti-quaving drug can turn the gene off. It's downregulated in the presence of anti-quaving agent. For most of these cell clusters, those are neurons. So we use iPS-derived forebrain neurons for the subsequent functional genomics, which I will show you in just a minute. But before we look at data, you might want to ask me, are those data reproducible? So we generate another set of brain organoids doing the same thing, and we found the same results. KHNYN highly expressed in virtually any cell type in the brain organoids. CBLN3, the signal is poor. We use iPS-derived forebrain neuron for subsequent functional genomics to study the two genes we identified during the GWAS. And we found CBLN3 and KHNYN, once we overexpress these genes, the most affected pathway is related to interferon signaling, which is unexpected because these two genes is really understudied in addiction or neuropsychiatric disorders. Then we functionally validate these findings by using additional iPS-derived forebrain neurons overexpressed with these two genes. The red is overexpressed with KHNYN, the green is overexpressed with CBLN3. Those genes are involved in interferon pathways. After we overexpress these two genes, interferon pathway related genes are upregulated, and that is really striking. And even more striking, we found both cortisol and interferon gamma, interferon alpha concentration also went up when we overexpress these two genes. We found a positive correlation for both interferon alpha and cortisol. So that means these two immune-related pathways, they are interrelated, they interact with each other. What about acamprosate? That is the drug we use to treat our patients. And again, we look at the same thing. KHNYN and CBLN3 expression went down in response to acamprosate treatment. At the same time, we studied cortisol and interferon alpha. We found their concentration also went down in response to acamprosate. That being said, acamprosate has an anti-inflammatory property, which we didn't know before. To summarize, I have shown you how we use multi-omics and iPS-derived brain organoids to study alcohol use disorder, and drug response phenotypes. We begin with a clinical trial that helps us to identify what proteins associated with treatment response and alcohol craving intensity. We also pursue a series of omics studies to identify the candidate gene for research. We use iPS-derived brain organoids and single-cell sequencing technology to better understand the molecular insights. All of these takes a lot of time and effort. So what is the key message I like to deliver here? The drug is not just a therapeutic. They can also serve as a molecular probe for us to study the pathophysiology of the disease and the molecular mechanisms responsible for treatment phenotypes. Our focus is precision psychiatry. This does not take just overnight because in psychiatry, we are not just dealing with the brain, we're also dealing with mind. And genomics alone is not sufficient to identify biomarkers. This is why we're adding additional omics, trying to better understand the biology so we can identify a better treatment target. None of those studies would have been possible without funding support. So I'd like to acknowledge our funding support from NIH, especially NIAAA, NIDA, and Mayo Center for Individualized Medicine. Thank you so much for your attention. Please let me know if you have any questions.
Thank you very much, Dr. Ho. And if you have any question, and then just turn on your mic and then direct ask a question, or you can just post your question in the chat box. So my personal question is that why you isolate the HDS11B1? And could you explain in more general term why your fishing strategy is focusing on the inflammation, especially related to the heavy alcohol consumption?
That is a very good question, Dr. Kim, because we started with a clinical trial that gathered blood samples for omics AC. First thing we did was genomics, and we found nothing at all. So we turned our attention to proteomics. But among the proteomics platform, even just within all links, there are a lot of platforms we can choose. And why is that we focus on inflammation? That is because for substance use disorders, especially alcohol use disorders, it's well studied neuroinflammation is one of the key mechanisms that relate to disease pathogenesis. So that's why we look at inflammation panel. And within this panel, there are over 300 markers. When we look at HDS11B1, this is using an unbiased way to identify potential markers we can pursue mechanistic study. And we didn't really expect to see HSD11B1 because it's highly expressed in the liver. But our samples are coming from plasma. So this brings us back to the biology. AUD is a brain disorder, but it also causes a lot of damage in the liver. So this is not just a brain disorder by itself. That's why we begin with the blood-based assays that have the whole system, and then going back to tissue-specific manner to study the mechanisms. Did I answer your question?
Yes, great. So let's go to the chat box. And the first question is that when brain organoids are exposed to ethanol, are just placed in the ethanol? And then maybe I will tweak the little question again. And then just the alternative, alcoholism is not acute disease, but chronic disease.
Yes.
And how to control experimental setting, not directly put into the ethanol in the brain tissue, how we can just control this one as a cloning manners. I just altered the question a little bit.
Oh, this is really insightful question. So what you're asking is if we can recapitulate drinking behavior in real world using iPS-derived brain organoids or model system. And I would say for the drug treatment we do here in my study, we do a week treatment. For example, for alcohol exposure, we use a certain concentration that is within heavy drinking blood drug level. And we do a week treatment with daily drug and culture medium renewal, trying to recapitulate chronic alcohol drinking. But this is not perfect because, number one, those patients we recruit in the clinical trial, they have been drinking for decades. Some of them started at like really young age, at age of 10 or 13 until adult life. So it is difficult to really accurately using what's being used in the real world and apply to the cell culture system. So for cell culture system, usually we will do a week or two ethanol exposure, and then we consider this is a chronic treatment. For acute treatment can be hours, but it's rare because it's never been the case. And can we really say this is how we define it's a perfect model system to study addiction? I don't think I can claim this, but at least you can justify why did you do this? Because we're doing pharmacology research. Every time when we do drug treatment, we have to justify what drug treatment you use, how often do you treat, and how much do you treat. So I don't have a perfect answer for this for sure, but I can provide my justification just for you to see this is a real challenge in our field because there's no standard.
I have a question.
Oh, yeah,
One
go ahead.
question. Yeah, there's been, you go an increase in popularity. I would say if you go to any bar or restaurant nowadays in non-alcoholic beers, non-alcoholic wines, have you or others been looking at non-alcoholic beverages in the setting of whether that's HSD11 and craving intensities or cortisone to cortisol activation in these non-alcoholic beers and how that can impact someone that struggles with alcohol use disorder?
We haven't got a chance to look at this. That might be because our clinical trial data is quite limited. That doesn't include this type of information. But certainly this is quite an interesting field to explore.
Yeah. Okay, cool.
Hey, go ahead.
Sorry, Keita, one more question because this is super interesting. And I think actually Shane alluded to this a little bit in his question in the chat as well of whether iPSCs would be a good kind of model for this, right? And I was wondering if you've noticed any, or if you've done the work of having, reprogrammed cells from patients of kind of like known outcome. Patients that have had or have dealt with alcoholism to a certain degree, and whether that is maintained to any extent after reprogramming, right? We know there's this complete erasure of most of the genes, but could there be some epigenetic signatures that would help in this case as well with an iPSC model?
This is really good question. We did study this using the same study cohort, looking at gene called FGF21 that is associated with alcohol use disorder. And that is also identified through our proteomics informed GWAS study. And we did find epigenomic mechanisms that are responsible for FGF21 levels. And that also correlate with catecholamine metabolism. So that study is a very comprehensive study using the same statistical and the model system to demonstrate how we identify the signal, what it does to the gene and the protein, and subsequent epigenomic mechanism. So a lot of people say you use iPS cells, you wipe off all the epigenomic profile.
Mhm.
How do you study epigenomics? But again, we can still do that if we use drug as a molecular probe. So again, that study, we expose those iPS-based models with drugs like alcohol, FGF21, or other compounds to study the drug signature instead of just epigenomic based.
Oh, this is super cool. Thank you for sharing the really, really cool work.
Thank you so much.
Okay, let's go to the chat box, and Dr. Ho, Ming-Fen, you can look at the chat box too. And Alex Weber, why do you think your secondary organoid had astrocyte versus astroglia in the first organoid?
So for the organoids data we use, it's 90 days after differentiate. These organoids, they do have astroglia and astrocytes, but the ratio is slightly different over time. The composition of these brain organoids, including neuronal cells, glial cells, progenitor cells, depending on how long we differentiate, the composition is different. And that's why we fix our study at day 90. That is similar to like second trimester brain age. But we found in the presence of the drug, we can still study the biology related to the disease by itself. So that's why we do this.
Yeah, so this one is a general question because heterogeneity of the organoid culture system is a general key we consider. And in your new apparatus, do you think that heterogeneity is increasing or decreasing?
I think also depending on the study cohort and the technique people use, because heterogeneity can be the biology itself or relate to technical issues. So if we're not talking about technical issues, I would say everybody has its own molecular signature. So for us, I have iPS-derived cells from patients. I also have unhealthy, unaffected controls. Sometimes they do behave different. And that is related to the disease pathogenesis. For example, opioid addiction. We have published single-cell sequencing data using iPS-derived brain organoids from patients with opioid addiction and unaffected controls. They do have very different profile, and they respond to opioid differently. And then we identify the potential mechanism responsible for this. So this is why we barely study N of 1 project or N of 1 study. Our iPS cell line is a patient population. So we usually look at the we look at the cell population in a bigger cohort rather than just one or two.
Yeah, we will continue to ask a question in the chat box until 50 minutes, and then 7 more minutes, and then after done, and then we will wrap it up. But everyone can feel free to send the email to Dr. Ho directly if we cannot receive your answer continuously. Next question is Fernando Alvarez. At the early stage, it would be possible to add microglia to the brain organoid.
Yes, that is a really good question because currently microglia we generate in the laboratory do not have the brain organ we generate in the laboratory do not have microglia because it's from different resource. It was initially from UCLA. So we now build a protocol to differentiate microglia separately. It's a 2D culture and introduce those cells into brain organoids. So we call this microglia containing brain organoids, and this is a different level. So the brain organoids technology has become more and more complex. In the past, we only look at region-specific brain organoids, but nowadays we can assemble different brain regions. We can also put different cell types into the mix. So it is feasible.
Okay,
but it could be challenging.
Yeah. Next question is Jaira Gonzalez, and everything is just a thank you message. And then next is Theta5 is abbreviated name. And then would it possible to add endothelial cell to promote vascularity and help the nutrient and the oxygen supply to the center of the organoid?
I think this is really critical. And I have seen people trying to use chip-like structure to build a vascular structure in brain organoid. So again, this is also feasible, and several groups already build protocol to generate vascular structure in iPS-derived brain organoids. So I think it's feasible, but again, depending on how you do it, can be really difficult technically and needs to have a standard to characterize those cells as well.
Okay, next question from Kellan Schroeder. And Dr. Ho, what is your input on psilocybin? Maybe you can confirm the drug name in the chat box. Treatment for alcoholism.
Okay, this is a really good question because we are not just study FDA-approved medications. My group also study psilocybin, psychedelics. Nowadays, this drug is a hot area in both depression and substance use disorders. And this is a Schedule 1 drug. So not every lab can study. You need to have DEA license in order to use Schedule 1 drug. So my lab is one of very few laboratories at Mayo Clinic to study psilocybin with a focus on substance use disorders, not just alcohol, but also opioid. And we use iPS-derived brain organoids for single-cell sequencing. And then we also take advantage of the samples from our patients, the blood samples, because sometimes what we found in the brain organoids, our goal is to identify biomarkers. But it really takes a lot of time and effort to generate brain organoids. So as a biomarker, you want to have access, like routine access, like blood, plasma, that would be really harmful. So once we have a signal in the brain organoids, we go back to the peripheral and then to find if the similar signature is out there. So now we have a project focused on psychedelics, and that will be out very soon. And I wish I had the opportunity to share this next time. That is a really exciting project.
Okay, good. Next question is from Theta5. In the organoid culture system, did you notice any sign of the neurodegeneration, neuroinflammation with a chronic alcohol treatment? I suppose no neuroinflammation since the model system do not have microglia. That is an interesting question.
A lot of findings relate to addiction or neuropsychiatric disorders. I found neuroinflammation is the heart of addiction because everything we found, either from peripheral or iPS-based CNS model system, neuroinflammation is always on the top. And it doesn't matter if we have microglia or not, we can still find those immune-related pathway show up, because microglia, they don't work alone. They also interact with astroglia. They also work with neurons or other cell types. So without microglia, brain organoid has many, many cell types, brain cell types. They still have functions related to immune or neuroinflammation. For example, ER stress. ER stress is one of the immune-related pathway keeps on showing up in both alcohol addiction and opioid addiction. And this plays a really important role in the pathophysiology. So I found microglia is important for sure, responsible for neuroinflammation, but other cell types also can contribute, and they might also play a very important role in the pathophysiology of the disease.
Okay, great. Now is 50, and then depending on your time zone, Pacific is 10:50. For some of them is from Europe. So therefore, their time zone is different, but this one could be my last question. Question from Sung-Yeon Kim, Sun-Young Kim. And thanks for Dr. Ho. Don't you think that alcoholic damage to brain organoid should be tested with acetaldehyde also?
Acetaldehyde is related to the pathophysiology, but it's no longer a drug target since disulfiram, the FDA-approved medication, is target on aldehyde. It works really well, but the side effect is really severe. So it's no longer a druggable target for alcohol use disorder, but certainly can be tested in brain organoids, but doesn't have much clinical implications.
Yeah, thank you very much, Dr. Ho. I really appreciate your participation, and also thanks for Tom and Omar for excellent and insightful lecture today. As discussed, while some of the example of all of our lecture series from Thermo Fisher is based on the Thermo Fisher product, but principle and the approach are broadly applicable across the similar platform and the system. And all lecture contents will be distributed through the email and also uploaded to the center YouTube channel after course is completed. And the next week, we will cover advanced feed-free human pluripotent stem cell culture, including optimization strategy and more detailed workflow. And this session will build directly on today's contents and then provide the deeper critical insight. And then also we will also discuss some of the questions we did not answer, unusual tissue culture behavior, depending on the patient-specific mutation. And Omar and Tom, do you have anything remark? No?
No, just a big thank you to Ming-Fen for a great talk as well.
Yeah, me
Thank you.
too.
That was excellent. Really
Yeah,
enjoyed
thank
that.
you very much. See you next week.
Perfect. Thanks,
Thank you.
everybody. See you next week.
Appreciate it.